Okay, so I'm going to go pretty fast. I'm hoping to sort of close out most of this chapter. Having said that, again, this is the chapter for anybody going into the medical fields that is the least helpful, least useful in your actual career and your later training, okay? It's incredibly helpful to have it in your back pocket, look it up, but it's not something you're ever probably going to have a lot of testing on or anything. I might go through pretty quick, but what I would like, and I'll probably not stop and say, do you guys understand, and did I lose anybody? I'm just going to expect you to, if you get lost, please let me know, okay? Even if you realized you were lost later in this class, just put a star or something next to it and go, email Dr. Clausen, you lost me here and I need something. Okay? All right. We can discuss it, okay? And we can put out some sort of supplement. All right, so this is actually what a high-performance liquid chromatography looks like. And you can see it's actually time-resolved. What you would notice is that we actually have a visible light, UV light spectrometer that's triggering every time a protein crosses it, okay? And so this is essentially just a trigger saying a protein crossed, okay? And that's what you're seeing here. You're seeing spikes. Every spike is probably a protein. The exact amount of protein may not actually register according to that spike's height, okay? Because it could be how well it absorbs. Now, you'll see there's a bit of tail ends. There's always tail ends on all of these because it'll always come off a little bit slower and you'll have little bits that don't quite release as fast, okay? The other thing you'll notice is the high-performance liquid chromatography, you could actually separate this. You could separate these to some degree, but not perfectly, right? And then you can separate this to a pretty decent degree, but again, not perfectly, okay? And then this you could almost separate completely. There is going to be some overlap and some impurity, but if you could run it again, you could probably separate it almost perfectly. Now, these separations, if you were to look at a traditional chromatography, you may only be able to separate 1 in 5 to a limited degree. Okay? Not completely. That's the sort of differences you're looking at. You're usually looking at a 10 times to 100 times difference, even if you're not doing it perfectly. Okay? And a lot faster. Notice how fast these come off. 5-10 minutes, right? Okay. Traditional chromatography can take 30 minutes to an hour would be a normal chromatography. Okay? They can take 4 days, depending on how well you pack that column, right? And how dense it is, how many interactions. Okay, so gel electrophoresis. Do you guys do gel electrophoresis downstairs? Yeah? Fantastic. I'm very glad. Yeah, I've been getting mixed messages from my students. I'm glad that they do. Yes, it is a much more advanced, much more finicky technique. And they don't actually have you guys make your own gels anymore. Yeah. They used to have us make them, but it's toxic and anyways, the electrophoresis is going to separate mixtures of molecules using a net charge that have a net charge by applying an electric field. So if you have charge on your molecules and you apply an electric field and put the molecules in the middle, what's going to happen? The positive will go towards the negative end of the field, right? And the negative ones will go towards the positive end of the field, right? And so if you do this, they're going to start separating based on their charges and their weights, right? And if you have a gel, they may also separate based on size as well, okay? So you're going to use this to separate proteins and nucleic acids. This is going to be carried out in gels. This is why it's called typically gel electrophoresis. Though you can do electrophoresis without a gel. Okay? There are 2 parts that can sum up to a technique, but you can do other electrophoresis. Okay? These are going to act like a molecular sieve, and i.e. they're going to make it more difficult for bigger things to move around, right? And so they're going to enhance that separation even further than just the base molecular weight would have, okay? then the small molecules are going to move quicker than the large ones through the gel. And so what you'll get is, like, if you want to eliminate all your amino acids, most of those are going to end up pulled out, right? All the charged ones will. All the neutral ones will just end up in the middle, okay? Because they're way smaller, way lighter than all your proteins. And then you can separate your proteins based on sizes and charges in the gel. By the way, what happens if you have something with like a 5+ charge versus a 1+ charge? If they had the same molecular weight, same size. The 5+ charge will move towards the negative end much faster, right? Yeah. And so what you typically get is a dispersion through the gel with little bands. You would put UV light to it. Do they use UV light downstairs? Okay. You can also impregnate the gel with fluorescent molecules, which would make them show up under regular light. Yeah, probably. That's a much easier way. Okay, cool. That's a great lab now. I'm really glad. The UV light is easier. If you put UV light to most proteins, they'll fluoresce themselves. However, you can actually put molecules that will attach to the proteins and be visible under visible light. Okay? But that's much more expensive and more toxic and more limited and more like, yeah. The UV light's usually just the better way. By the way, that's actually what you were seeing here too, is this UV absorptions that were happening. And a lot of those will just fluoresce back out as visible light if you do that to a protein. So here's actually what we're seeing here. And in this case, we're only developing it in the positive direction. Can you see that? The negative direction is just getting pulled off. Okay? You can develop them in both directions. You'd have a flat gel or a gel that's like this, and then you would have a positive and negative end on both sides of the gel. Okay? But by doing so, you half the amount of distance you can develop it, right? In either direction. And so most times you'll see them develop in only one direction. You can flip it if you want. All you have to do is just flip the polarities. It's really easy. But yeah. So this is your porous gel right here. This is going to be the direction of electrophoresis. The negative things are going to move downwards. The positive things are going to move off the gel into this solution over here and stay there and be left behind. Okay? Typically, this will be some sort of buffer solution with the proper cations and anions, as in like salts in it, because you do not want to mess around with pH on your proteins, right? They'll unfold, denature. Yeah. So you can see here, smaller stuff moves down faster overall. Medium, large doesn't move a lot. Okay. This also, though, is resolved with charge. So it's a size versus charge difference. Right? Do you guys remember your MS, GCMS, mass spectroscopy from organic? This is very similar to mass spectroscopy, just mass spectroscopy does this a hell of a lot faster and in a gas phase, right? Yeah. Though it would not work well for proteins because they can't go in gas phase. They weigh too much. So right here we actually have a polyacrylamide gels. These are going to be highly cross-linked, and this is that cross-linking that is creating that maze effect where things can have trouble getting through. Okay? Without the cross-linking, you'd get tunnels in it, and so you wouldn't get good separations. You'd get very wavy effects. Okay? Okay, so this is actually the polyacrylamide. They used to have you make this, but these chemicals are somewhat toxic. When you actually combine them, they're not really because they're film, so you can't breathe them and stuff. But yeah, so they don't really have you make these anymore. But yeah, anyways, you get the idea. And there's big corporations that make these many, many times and can do it really well, better than you could do. So honestly, it's a better way to do it. This is SDS page. SDS page being sodium dodecyl sulfate, polyacrylamide gel, electrophoresis. That's the abbreviation of all that long thing. This allows the accurate determination of mass within your gel because it's something with known mass, known size, and known charge, right? And so what you can do is run this alongside all your other stuff, and this will give you an idea of everything that is around that band where this comes out in your gel is around that size, isn't it? Well, size versus charge. So for most proteins, 1 molecule of SDS will bind for every 2 amino acids. I'm sorry, I got ahead of myself. This is actually the one that binds to the amino acids, excuse me. I wasn't paying attention to where I was. So let me back up here. This is not an internal standard, which is what I was talking about before. This is the one that's going to bind. This is an anionic detergent. You can see it looks a little bit like soap. See the polar head, the non-polar tail? Yeah. I guess I wasn't really paying attention to what page I was on. And so what's this going to do is it's going to denature your proteins partially. If you denature your protein, what it's going to do is unravel. This allows you to separate things that would be otherwise like globulins, right? So globulins all have kind of the same size and shape, right? And they don't change a lot depending on how much of the amino acids are in their exact globulin you have, right? And so if you partially denature these, this is going to allow you to stretch them out, and then based on their size, they'll tumble through and get caught, okay? So it'll allow you to more accurately resolve these different bands of sizes. So proteins, for most proteins, 1 molecule of this SDS here is going to bind for every 2 amino acids, okay? So for every 2 amino acids you have in that protein, you'll get 1 molecule of this SDS, typically. The proteins are going to have the same charge to mass ratios and migrate in the gel on the basis of mass only when you use this, okay? And so you can resolve that issue of you'd have, things with like a 3+ charge could be 3 times the mass and end up at the same place, right? You can resolve those issues, okay? All right, so these are going to be proteins that are separated by SDS page. We have here a dye that we've dyed the gel with. Usually you take the gel, you run it, you would then put it in a dye. The dye is then going to stick to the bands of the protein, though you don't necessarily have to dye if you use UV. Did you guys dye at all? Did you? Okay. And then you also had to use UV to show it? Okay, yeah. I mean, it depends on the dyes. The dye plus UV will make it easier to see. And you don't have to use such a powerful UV source, which helps. Like I have in the lab? Yeah. We do something similar with this lab we'll have, where we're going to have fluorescein impregnating our separation, and then we'll use UV to cause that to fluoresce. So here we have the different lanes here. And these are going to be measured in kilodaltons. Remember, kilo meaning 1,000, dalton meaning basically an AMU, but biology, right? Why they had to make up their own fucking term, I have no idea. Whatever. So here we have different kilodaltons. Notice the 150 is up here. And then as we get progressively further down, we would have lighter amounts, right? Okay. Anybody have any questions about that so far? I know I said I wasn't going to do that, but... All right. Okay, so electrophoretic mobility of many proteins in SDS polyacrylamide gels is going to be linearly proportional to the logarithm of their mass. Now, you got to keep in mind that, so right here we actually see a plot of their mass versus relative mobility. This relative mobility is the fraction of the total amount you could have went, okay? So you would have a leading edge here. If you developed it all the way to the edge, it would be the edge. If you didn't develop it to the edge, you'd have to make sure you mark that out some way and know that, okay? Typically, it would have some little lip on the container that this would like dig into the gel, and then you would have a little line there, okay? And then you would stop developing when you hit that line with your leading edge, right? Many times your leading edge will actually have something in it for these, something light. And so this is probably the leading edge right here, I'd imagine, but it could not be. I can't see that well enough to see the lines or anything. So the relative mobility is going to be the total amount it could have traveled, will be the denominator, and then what you actually did travel for that band would then be the numerator, right? And you'll get a fraction, and that is this fraction right here. Note that by doing that, you actually are making this so it is a percentage-like relationship, right? Okay. Meaning like the last 5% of a percentage is a hell of a lot more than the first 5%, right? Everybody understand that? Okay. Okay. These then are just straight up masses in kilodaltons. This right here is then monitoring the process of this. And so here we have a homogenate. Remember the homogenates we talked about where we just broke everything up and we have everything in there? That's a homogenate, okay? And this is what it would look like in a gel that we have developed. This right here would be a salt fractionation. Remember we do the salt. As we change the salt concentrations, different fractions will salt out. Okay. That's what that looks like. See how we've salted out some of the components? And some of them, notice this line here, where it's darker, lighter. We don't typically get in a salt fractionation 100% removal. Okay? It's very rare to get 100% removal. Notice mostly the lines just got lighter, right? This is going to be ion exchange chromatography. You can resolve things enough with ion exchange chromatography that you can actually eliminate them. See how we eliminated that band? Okay. Now, sometimes you won't be able to eliminate completely. Well, this one we probably eliminated several components that were in that band. Okay? Because it's pretty dark, but it's just narrow. Right? But other places, we just sort of lightened it, right? Which means we probably didn't eliminate that completely. We only partially eliminated it. We couldn't resolve it completely. This right here is the gel filtration chromatography. Notice these we basically completely eliminate some of them. Okay? Okay. Notice the affinity chromatography here. Everything else is gone. Now, we will get a broadening of the band because oftentimes you'll have bound things to it now. And so the amount of them that is bound to it at that given moment is going to broaden that band, right? And you've added other factors that will broaden the band. Does that make sense? Okay. Okay, so the isoelectric point, pi, this is the pH at which a protein has no net charge. The isoelectric focusing is going to separate proteins in a gel based on their pi. And so what you'll do is you modulate the pH so that you can make different proteins neutral and different proteins be, depending on their side chains, right, be positive or negative, depending on which way you're going. Whichever you want, really. Just change the pH in one direction. If you have a mixture of proteins that's placed in a gel with a pH gradient, each protein is going to migrate to its pi because it'll go to that point. Once it hits that point, your electric field can't react on it anymore, can it? Okay. And so what you typically have is you have a middle point that is some pH, okay? Okay. And then you'd have a gradient that slowly goes to different pHs. Right? And you can fairly easily do this actually by taking two different buffers, putting them on the different sides of the gels while it's setting, and allowing them to impregnate through. Okay? And what you get is that separation between them. All right? Okay. Anyways, that's why you'd make your own gels and stuff, by the way, is because you can impregnate the gels with different factors. Different salt concentrations, if something's precipitated out with certain salts or would become neutral with them. You can impregnate them with the dyes in the first place or binding molecules to make things heavier or bigger. You can pregnate them with different pHs in a gradient, okay? Which you could do the isoelectric focusing. You get the why that's isoelectric focusing. You're going to focus each population on different isoelectric points. Remember that added the protein's PI. So every protein is going to have a PI where it will be neutral for that pH, isoelectric point PI. It's going to have an electrophoretic mobility. Its ability to move on that plate then, that gel, is going to be 0. Okay? You'll end up stuck there. Again, this is a figure of this happening. And so notice that it actually doesn't matter where the hell you put the proteins. Okay? You could scatter them all over the damn gel. Doesn't matter. All right? All that matters is that you've put a field across this with positive and negative M on one side and the other, right? And so if you have a low pH side and a high pH side, it will migrate to wherever its PI is. Doesn't matter if it starts in the left or right of it. Okay? Which is pretty cool, actually, because it makes this really robust, except for the fact that you can actually affect the protein structures. But if you're using things like SDS already, you're already denaturing the proteins, right? So it doesn't matter. Okay, so again, notice how we focused all the red protein here, all the blue one here, all the yellow here, all the orange here. Yep, there we go. Despite the fact that they started all over the place. Okay, so two-dimensional electrophoresis is going to separate proteins in two different directions. The isoelectric focusing in a horizontal direction. In the other direction, the vertical direction, perpendicular, it's going to be SDS page. So this is going to yield a gel with proteins that have a separation based on their PI in the horizontal direction and their mass in the vertical direction. Whoa, huh? All right, this is what this looks like, okay? And so what you have here is on the top, you have a isoelectric focusing that's going to occur. And then you would have a reservoir that you can then release, the SDS page, once you have that basically done. And then the SDS page will come in, denature your proteins, add every 2 amino acid like we talked about, and then they're going to get pulled downwards. Okay? Typically, this one's just going to have one charge applied. All right? Did that make sense? Okay. This is what they actually usually look like in real life. Okay? You'll see that there's going to be a lot of like starburst pattern sort of stuff because you won't get perfectly resolved on the top because this is real life. Proteins are going to have, things bound to them and be a little bit different. Okay? You'll never get a perfect, straight, beautiful band. It's always going to have a bit of blur to it. Okay, so the two-dimensional electrophoresis, this can detect differences in protein expression under different physiological circumstances. If you do a careful enough and good enough job of this, you can separate pretty damn similar proteins that have very, very, very little difference, okay? Based on very fine differences in their structures and physiological circumstance reaction, if you will. How their PIs, how they change due to various different conditions. You can actually do a lot with this, more than even I'm talking about. I can't even. This could be like a damn class. All right, so the effectiveness, I didn't actually know that much about this. I'm not that good at this technique. I did it a few times, but I knew somebody that did this for like 6 years straight and was a master of this stuff. They could separate variants of the same protein using this technique. It was ridiculous. The effectiveness of a purification scheme is going to be measured by calculating the specific activity after each separation technique. And so you can actually evolve your separation technique to then better target the proteins you're looking for, okay? And so again, the specific activity is the ratio of the enzyme activity to total protein concentration. It tells you how much of that protein is that enzyme you're looking for. Or it doesn't have to be an enzyme, it could just be something that binds to it, but you get the idea. Enzyme is more common. So this should increase with each step of your purification because more of your protein is going to be removed while the active state is the one you're purifying, right? And if this ever goes down because of your purification, what do you know? I done and screwed up, didn't I? Yeah. That means that I've lost some of my protein in the active form. Okay? Now that could mean that I've denatured it, it no longer works. And I couldn't get it to fold back, okay? But yeah. So again, should increase with each step of the procedure because you're going to be concentrating that protein as opposed to the other proteins. The SDS page actually allows you a visual evaluation of your purification scheme. You should see a band that corresponds to the protein you're looking at. And what should happen is all the other bands fade away, and that band mostly stays the same coloring, same size. Okay? Okay, so again, this is that quantification of what we just saw in a visual representation where we have the different steps we can do on the purification process. Okay? Okay. And notice that again, we get our percent yield is going to go down all the time because we're going to lose more and more of that enzyme every purification step. Just like an organic lab, right? Okay. But what's going to happen is our specific activity will go up because all the other proteins will get lost more, right? Okay. And our purification level is going to go up because we're purifying at every step. Okay? Note that if you can't get a very concentrated amount to start with, that you may have to stop a purification scheme only halfway through because you would lose so much percent yield you can no longer get what you need, right? Like what if you needed 50% of this to do the remaining tests? Can you do this last purification step and expect to be able to do the test? No, you're just going to have to deal with that, okay? That happens a lot. Something you need to keep in mind and track of. Ultracentrifugation, this can be used to analyze the physical properties of biomolecules like mass, density, shape, and interactions with other molecules. The sediment coefficients are going to be the quantity of the rate of movement when exposed to centrifugal force. This is going to be expressed in Svedberg units. You can tell we were walking into the biological realms, right? Because everything's named after people. But yes, Svedberg units, the smaller, it's a capital S. The smaller the S value, the more slowly a molecule moves in centrifugal field. Centrifugal field is just downwards in the centrifugal tube, okay? And if you have a large value for the S value, this is going to cause that to end up on the bottom in a pellet. If you have a smaller value, it'll be much harder to move it to the bottom of the pellet. If you have a small enough value, you're never going to get it down into the bottom of the pellet no matter how much you spend. Okay? At least not with something not the size of a football field. Right? The more massive particle is going to sedimate more rapidly than does a less massive particle, as you probably realized. Elongated particles sedimate more slowly than the spherical ones. Spherical ones that have the same mass are going to have less, like a ship. Okay? Why is a ship not like a skyscraper, pointing up and down? It'd sink, right? That means it's sedimentation value, right? Because everything on the Earth is undergoing something analogous to centrifugal force, which is called gravity, right? Okay. But if we get something that's, a law of space that can tumble and take up more volume, then its sedimentation value is going to go down, isn't it? Okay, because it's going to get harder for it to move through the solution. It'll float better. Okay? Okay, so this right here is the S values, Svedberg units, the molecular weights of some very common proteins that we would normally see in samples. God, I love this book, how it just references publications on everything. That's real. They're actual values. Yeah, so you can see here the different Svedberg units. Overall, the Svedberg units typically follow the molecular weight. But again, if you have elongated structures, the Svedberg units can be lower, okay, than they otherwise would. Very compact globular structures are going to have higher Svedberg units than otherwise. Okay. Recombinant DNA technology is going to afford several different advantages in the production and purification of proteins. So we can actually, remember how I said you got to start off with enough protein to reach the end and actually have a useful amount, right? Okay. Kind of like our caffeine lab. Remember caffeine lab? I like 10 times the amount you're supposed to use. Because by the end of it, you lost so damn much that you had almost what? 20 micrograms? Something like that. Similar here, we're going to lose 65% of our sample if we do this entire purification scheme normally. Okay? We may even lose more than that if it's something hard to deal with. Right? And so with genetic recombination, recombinant DNA technology, we can actually change how that protein's expressed and have it where it's produced in much, much, much larger quantities. We can go up to like, the lab I used to work with, they produced it in 10,000 times the quantity it would normally be produced. Okay? Yeah. And they had to. They even struggled getting it then. Each sample was worth over $10,000 when they made it. So the proteins, again, can be expressed in large quantities. These proteins can be then modified with affinity tags. This will allow the purification of the protein or to visualize the protein in the cell itself, which is really cool. That's how they make those pictures, by the way, where they can visualize like where the different protein markers are in the cell. The proteins with modified primary structure can then be generated. And so we can actually change that primary structure and see how putting a different amino acid in that spot changes its activity and changes its structures. Okay? So the antibodies, these are going to be proteins synthesized in response to the presence of a foreign substance, is going to be called an antigen. The antibody is the protein synthesized. The antigen is the one that the foreign substance is that's going to be targeting. Okay? In case I wasn't clear on that. Epitope is the antigenic determinant. This is the specific group or cluster of amino acids on the target molecule that an antibody is going to recognize. So your antibody is going to have that variable portion that's going to recognize and bind to an epitope. And that will be the antigenic portion or determinant. The antibodies to a specific protein can actually be generated, of course, because we can actually cause like the easy way is to usually just sort of flip the dice really fast. So what we do is we just modify a bunch of different ones, have a cluster of stuff that's all modified and has that high variability loop, very, very variable, and then we see which one stays. Okay? And remember how we said you could attach anything to the specific targeted columns? Guess what we would attach to the specific targeted column? The epitope. Yeah. If it binds to it, it'll stay on the column, and we can release it individually, can't we? Yeah. Anyways, polyclonal antibodies, these are heterogeneous mixtures of antibodies. These are derived from multiple antibody-producing cell population. Each antibody is going to be specific for one of the various epitopes on an antigen. And so right here we actually see a polyclonal antibodies. Here we see an antigen right there. These are our antibodies, of course, that we're attaching to it. And you can see these are all targeting different parts of this. Okay? And notice that we typically have each of these antibodies here targeting one portion, but we can actually have them cross-linked together. Okay? Okay. Now remember, that's that loop of 6 we're talking about, okay? Remember, this is the head here with the two sides. This is that body that's going to get released. Okay. And each of these has the 6 hypervariable loops. Okay. So those hypervariable loops are responsible for binding to one site on there. But you can have multiple ones that bind to different parts of the antigen. Monoclonal antibodies, see the difference, poly, mono? Mono meaning one, right? And so monoclonal antibodies are going to have identical antibodies that are produced by clones of a single antibody producing cell, and that's going to be this right here. And they're all going to stick to the same spot because they're mono one, right? They're going to be targeted to bind to one part of the antigen, or otherwise known as a epitope, right? All right, so immortal cell lines produce monoclonal antibodies because obviously they're not going to change over time much. We're trying to keep them immortal, fixed, same, not changing much. These are generated by fusing normal, short-lived antibody-producing cells with immortal cells from a type of cancer called multiple myeloma. Yes, all immortal cell lines are cancerous cell lines in some way, okay? Because they're not accepting kills, right? They have to. If they were accepting kills, they wouldn't be immortal. They'd die, right? Okay. And yes, your cancers, many of your cancers would survive you if they kept getting nutrients, in case you're curious about that. This is going to result in a hybrid cells called hybridoma cells. Literally, hybrid, and then the oma. So, yeah. The monoclonal cell line is then going to be isolated by screening for the antibody of interest. If it doesn't have the antibody of interest, then we look for another one, right? Okay, and this is actually how you prepare the monoclonal antibodies. You take a living organism, like for example a lab mouse. You inject it with the antigen. Don't inject it all at once usually. You do it over time, usually a regimen. And then once it starts expressing antibodies against that antigen, you're then going to collect the spleen cells. You'll put them in with myeloma cells. This is a cell culture, an immortal cell culture myeloma line. It's a pretty standard thing. You can buy them from industry. Though there's a very limited number of them in the US that are allowed legally. Okay? There's a lot of laws involving them now. Anyways, they will then fuse in a polyethylene glycol. The polyethylene glycol is going to loosen the cell membranes, causing them to, when they come together, fuse. Okay? And see how the cells are literally fusing. This is going to allow you to end up mixing the internals of the cells. I.e., they're going to start taking on all the RNA, DNA, cell membranes, etc. of the others. Okay? And so you're going to select and then grow hybrid cells. These selected cells make antibodies of desired specificity. Literally, usually you do like a bunch of these. You select little targets, you grow them out, and you see if it's producing what you want. And then the ones that are producing what you want, you keep. All the other ones, they go in the autoclave and die. Okay? Actually, how that AI is done too. Weird. So, anyways, you select the antibody for the antibody that you desired for that specific specificity. Then you propagate the desired clones. You'll grow those in mass cultures to produce your antibodies. You can also then inject them into mice and produce tumors to see if the antibody grows viably in an organism. Okay? And you can actually then modify that. Yeah. If they're in the myeloma line, to ensure that they're actually producing the same antibody, you can resolve it against the mouse's antibody. And you can inject it, but because it's a cancerous cell line, it's going to produce a tumor. Okay? Now that doesn't actually mean the mouse will die, but it's going to die because you're going to kill it when you're done. But, you know, most mice will have tumors of some sort. Like, that's very common in nature. Hell, look at humans. It's very common in humans, right? Having a little tumor here and there is very, very common. That's why if you ever they never do full body scans. The reason they never do a full body scan is every human has some tumors. Okay? They're benign, they're little microtumors. They don't mean anything. But if you did a full body scan on every human, you'd have to do like 10 surgeries on every person. Okay? So they don't do it. The antibodies can be used as reagents to quantify the amount of protein or other antigen. Antibodies are amazing science, by the way. You can use them for a truly mind-boggling array of things, but again, they can be used as reagents to quantify proteins, so you can literally make an antibody that would then recognize the protein you want as the antigen, okay? And then you could have it target it and literally bind to it, and then make it into a purification column. Okay? You see how these all fold back on themselves? And now you're having tools that you can do real science with, right? Okay. The enzyme-linked amino-absorbent assay, ELISA, quantifies the amount of protein present. The antibody is going to be linked to an enzyme, like horseradish, for example, horseradish peroxidase, that's going to react with a substrate that will produce a colored product. And so this is a method of using this same technique to produce an assay method. Okay? This is actually a very common assay method now. Like 20 years ago, this was like cutting edge, millions of dollars to do this once sort of crap. Now you buy them for like a couple $100 and they even have them where they'll automatically do it on a blood sample. They have like a little computer chip with them in it. Yeah. You can buy them for 25 cents to check if you have HIV. Yeah. Really wild. Really progressed. This right here is the indirect ELISA, and then we're going to talk about the sandwich ELISA. I know, sandwich. Okay. So this is the indirect ELISA. What's going to happen is you're going to have an antigen-coated well. You'll then wash that, and you'll have a specific antibody that will bind to that antigen. Now, because the well is coated in the antigen, the antibody will then bind to it, right? And that will be fixed and bound to it, at least for now. We'll then wash that to make sure any of the excess comes off. Okay, that's what the washings are for. And then you're going to have an enzyme that linked antibody that will bind to the specific antibody that binds to the well antigen. Okay? So you have an antigen you produced. It's going to bind to that antibody, that binds to that antigen that coated the well. You then have an antibody that binds to the first antibody. Okay? And then what's going to happen is that antibody you had connected to an enzyme. Okay? You wash it to get rid of any excess. Then you're going to add substrate. This will then be converted by enzyme into a colored product. That colored product is going to tell you how much of something you have. And so then you can measure how much of the enzyme you have, and thus how much of the antibodies you had, and thus how much of the antigen was bound as well. Okay? So if you want to measure the enzyme, for example, you could measure the enzyme by having an excess of all these other things. Right? Then the enzyme will be the limited one. It will tell you how much is there by how fast the reaction occurs. Right? Okay. And again, that color formation, the speed of the color formation is going to be proportional to the amount of that specific antibody right there. Actually, you can do it for any of these coming down. You just have to have your limiting one being the one you're measuring. Okay? The sandwich ELISA, this is going to have a monoclonal antibody coated well instead of an antigen coated well. See how we're starting with the antibody coating the well instead? Okay. So you're then going to wash any excess off. The antigen will then be introduced. This will bind the antibody. You will then introduce a second monoclonal antibody that linked to an enzyme that will bind the antigen that you used before. And so it will then bind the immobilized antigen, which is why you had to wash here, by the way. And that will leave you only with, once you wash again, your enzyme-bound complex. Okay? And then what you can do is introduce substrate. Again, that reaction rate is going to tell you the concentration of your limiting component here. Okay? Okay. Oftentimes you can do this for the antigen, is a very nice way to do it. Figure out how much antigen you have in a given sample. But you could also do this for the enzyme as well. Anybody have any questions? Okay. You don't normally do this for the coded well part, by the way. That'll be very difficult to make sure you have limited in one and measure it in a way that matters. Okay? But it could be done, probably, theoretically. How's my time looking, by the way? 5 minutes? Okay, we can get a little bit more time. Primary antibody, this is going to be an antibody specific for the protein. Secondary antibody is going to be an antibody specific for the primary antibody shapes. Okay? So primary antibody binds specifically to the protein itself. Secondary, second one that binds to the antibody that's primary. Okay? This is going to be attached to an enzyme that generates a chemiluminescent product or contains a fluorescent tag. This enables identification and quantification. Western blotting. Did you guys do any Western blotting? Yeah? Just talked but didn't do it? Okay, that's fair. That's fair. No. So Western blotting, we'll talk about again. Are proteins that are separated in an SDS page gel. They're going to be then transferred to a polymer. They'll then be stained with a primary antibody. Again, the primary antibody right there, the one that directly binds to the protein. This is then going to be stained with a secondary antibody that will bind to that primary antibody. And then you can quantify it because it has a fluorescent tag, right? And so you can see it. Okay? All right, so this is actually Western blotting right here. Here we have our gel. We'll then transfer our proteins to our polymer sheet here. We'll add the primary antibody. Remember, anytime you add these antibodies, you have to wash in between. If you don't wash the excess off, they'll also bind the next antibodies, antigens, etc. Okay? So the next one is going to be adding a labeled secondary antibody to this. Ta-da! And you'll do, again, another wash. Then you can then expose this to typically UV and see the line that shows up now. Okay? And that tells you where your protein is in your previous SDS polychromic gel. Right? And so now I know that band over here is the one. Okay? All right, so step one, for the co-immunoprecipitation that enables the identification of binding partners, step one is incubate and extract with a monoclonal antibody against a specific protein. Step two is add agarose beads that are coated with an antibody binding protein, like protein A, we're going to call this, okay? Step three is to separate the antibody-bound protein complex through centrifugation. So keep in mind that because we've added these agarose beads and they bind to that, the weight went up a lot, didn't it? And so what's going to happen to it when we centrifuge it? It's going to make a little pellet, right? And we can then resolvate that to get it back. So under optimal buffer conditions, any additional proteins are going to be bound to the original protein, will also precipitate as well with it. And so if we have a protein complex, we can get the whole complex this way. Okay? The identity of other bound protein partners can actually be probed by other methodologies. Right here we actually see pictures of this. So here we have a cell or tissue extract there. We add our antibody that's going to target our protein here. And so the antibody then attaches to this. We add proteins A beads. These protein A are going to be attaching to those antibodies. Okay? And so they attach the antibodies. They cause them to weigh significantly more now. And when we then slowly spin this, so we don't collect a bunch of extra stuff we didn't want to collect, this will be the heaviest component. Okay? And so we can selectively remove specific proteins, complexes from our supernatant that we got before without doing things like salting them out and stuff. Okay? Okay. I'm done with time, right? Okay. Right, you all have a wonderful weekend. Let me know if I lost anybody anywhere or anything like that. Damn, I didn't quite get as far as I needed to get.